Good evening, everyone, and welcome to the last session of this workshop. So we've actually come to a very important moment, which is the final lecture by Benoit Roux. So it is my great honor to announce this final lecture, which we all look very much forward to. And Benoit Roux will talk about unraveling the mechanism of C-type inactivation in potassium channels. Please, Benoit.
Unmute myself, yes. Okay, so let me share the screen. Application window. This guy. So do you see my PowerPoint now? Yes?
Not yet.
Oh, and if I do this?
Yes, we do. We see it now.
Okay, do you see the mouse if I roll the mouse on the screen?
No, I think there's a little pen at the bottom left of your screen, and if you click on that, it gives you the options.
Oh, yeah.
For the pointer.
Oh, yeah, I can write, you mean?
You can write, but okay, no. So that there should be other options.
Well, it's okay to write.
Okay.
So I'm very grateful to give this talk here, and I'm so disappointed from the quality of the lecture that I wasn't able to be in Italy. You know, I sure hope that we will all be able to get back together very soon and just like meet as we normally do in person. So inactivation. Of course, by now we're in 2021. Many people have seen structures of ion channels, and ion channels are primarily known for ion conduction and selectivity. But of course, at the biological level, these are like the two basic action of a channel. But there wouldn't be biology if there wasn't regulation. And by regulation means that you have to be able to turn on and turn off this process. So inactivation, if you look at this cartoon here, inactivation, this is like a current. This is the current here. And this is time. So when you give a stimulus, you get like a bump of current. And this would be observed experimentally on a population of channels. And that's the process is when you give a stimulus, you get a bump of current, but then it goes away. And this was observed for many years, for example, on voltage-gated, voltage-activated channels, where you give a change of membrane voltage and you'd see a pulse of macroscopic current. And then people like Francisco Bezanilla realized that this is inactivation, and inactivation is structurally related to a part of the protein that has been then named the ball and chain mechanism. And if you remove the ball and chain, then this fast inactivation was going away. So now you don't have the reduction of current, which was basically caused by the ball blocking the pore. But now you get what people call this slow inactivation. And the fast inactivation was called N-type in large part because it was a part of the protein that was close to the N-terminus of the protein. And so the other one by opposition was called C-type, as if it's close to the C-terminus. Of course, that's a bit silly because it's not close to the C-terminus, but the name has stuck around. So C-type inactivation is believed to be a gating mechanism that stops the ion conduction and primarily comes from some sort of conformational change in the region of the narrow pore. And there's many over the years, many effects are known to affect C-type inactivation, or sometimes called slow inactivation. It's, for example, the type of ions, like if you have rubidium instead of potassium, these are the inactivation will be less pronounced or slower. Of course, mutation close to the selectivity filter will affect inactivation, and also the concentration of potassium ion on the outside will affect inactivation. So when you study that, just to place your ideas, like imagine you have a channel that has the pore domain that's closed, you give a stimulus, and now the gate opens, and presumably the channel is able to conduct, And macroscopically, this is just, I'm showing one cartoon with one channel, but this would be, the current trace here is representative of what you would get from an ensemble, a population of channels. And then after a while, there's a conformational change of the selectivity filter that becomes non-conductive. And if you remove the stimulus, then you close back the gate. But for a little while, the channel would remain non-conductive, even though now the gate is closed. And so this diagram actually also gives you a way to study the kinetics of this silent process, because you can give back a stimulus at different times, you know, and try to see if you are ready to. Of course, if you wait a long time and you give the stimulus here, maybe you recover the full activity. But if you try to give the stimulus too early, then you don't recover the full activity. So you can probe this silent mechanism in that way. And in this cartoonish view, there's really two parts of a channel that affect the regulation. The first part is the activation gate. This is the intracellular gate that responds to an external stimulus. That could be ligand binding, it could be a voltage change, it could be a pH change. And so that's the activation gate that responds to an external stimulus. And then you get the selectivity filter itself, which undergoes a conformational change that is not under our control, is spontaneous. And you would have these four dominant states as a cartoon to represent the activity of a channel. When we got the first potassium channel, of course, the KcsA is very, this is a bacterial channel, but has a lot of homology to eukaryotic potassium channels, especially for the pore domain. And the crystal structure that we got at the time was clearly conductive, in the sense that it looked like ions could go through the selectivity filter. But nonetheless, it was possible, even though this is a bacterial channel, to show in macroscopic current, and this was done by the lab of Eduardo Peroso, that if you give a stimulus, and in the case of KcsA, the stimulus was a pH jump, you would get a macroscopic current. So this is a macroscopic trace with many channels. You get a peak current, and then it would decay. Here it's 3 seconds, so you can see it decays fairly slowly. And when you decay, this height is about maybe 10%, you know, of the height that you get at the peak current. So that would be like the sort of the depth of inactivation for a population of KcsA channels. And after labeling different parts of the protein with spin labels, it was clear that even though you get 10% of the current, the activation gate is still open, and clearly it must come from some other part of the protein. And later, Eduardo crystallized the channel in various states of openness, from 15 angstrom to very wide 32 angstrom. And as the channel is opening more and more, you can see that the filter is sort of undergoing a conformational change, where it appears that ions would not be able to permeate anymore, because there is a barrier in the middle. You cannot really permeate anymore. So this is sort of the paradigm for KcsA. It's been very frustrating, because there's now several ion channels have been crystallized, many bacterial channels, but also, you know, non-bacterial channels, eukaryotic channels, and the conformation of that we call, this is what we call the constricted conformation, has so far been observed only for KcsA. And it's not available for many of those channels, even though we have crystal structures of them. And so that became a kind of well, it certainly contributes to our confusion, you know. And so this is the plan of the talk a little bit, is that so far we mostly have direct information about this constricted conformation that would be putatively non-conductive for the KcsA channel. And I'm going to document that more. All the cartoons I had, like on my first slide, actually is the paradigm of voltage activated channel. Is in large part, this is data that came from the, you know, the shaker potassium channel, which is, or shaker-like, sometimes is MthK, also the big conductance potassium channels. But a lot of this inactivation was done with voltage activated channels. So it's important to go back to that The pore domain of shaker is very, very homologous to the pore domain of KcsA. So this one has, of course, voltage sensor, but the pore domain is identical to about 40% of the residue. So that shows that, in large part, what we learned from KcsA should be a message that you can carry over to shaker and other voltage gated channels. In the Herg channel, I'm going to end on the Herg channel because that's the channel where perhaps the inactivation is the most critical for a number of therapeutic and biomedical reasons. And so I'm going to end up with that one. So KcsA, this is a study we published just a few years ago where we were investigating the propensity of the filter to adopt this constricted conformation, but depending on the size of the opening gate. And essentially the process is that, okay, we write the cartoon where you open the gate and now spontaneously the channel would conduct for a while and then undergo this conformational change to an inactivated state, a constricted conformation. Is this really true? And in molecular dynamics, of course, you see that in long simulation, at least. So this is a process that is observed over, let's say, about 1 microsecond of hundreds of nanoseconds. So you can see, like, for example, in 600 nanoseconds, these lines, this is the distance that is basically across the filter. So when the distance becomes short, like 6 angstrom, that means the filter is pinched. When the distance is a little longer, like 8 angstrom, that means the distance is larger and the filter is conductive. And so you see here that, and of course you have 4 monomers, so you have 2 distances. So this is basically the conformation where the channel is constricted like that, with the distance is shorter, and you have two distances. And typically in KcsA, in wild type KcsA, the 2 distances will sort of collapse to a shorter distance about the same time. So it shows that there is a cooperativity, and this happens in about half a microsecond, basically. When we do this, there's always some water molecules that will populate some sort of a crack or a cavity behind the filter. And if you look at the density of these waters, they occupy always very precise site in the KcsA. And in fact, these waters were observed in crystallography by Rod McKinnon in 2001 already. And this is a movie where I think this is perhaps the longer trajectory, but you're going to see spontaneously you start from a filter that is conductive, it will collapse, and then the water will come in. So this is conductive, and the gray part here is the reference structure that is constricted. And you see, over a trajectory, the waters will just come in spontaneously and occupy their crystallographic position, and then the filter is now constricted, and it jiggles around this gray structure, which is the X-ray structure. So the molecular dynamics is really taking from one conformation to the other in about half a microsecond. There's no doubt about that. Now, staring at spontaneous transition is very cool, but of course, it's also nice to be able to have like perhaps a more statistical mechanical view of things. And so one way to characterize that is to look at potential of mean force. And so what we do is look at the potential of mean force as a function of the pinching, what we call the pinching coordinate. It's the distance between the 2 carbon alpha, the 2 glycine at the center of the filter. We often do this as a 2-dimensional potential of mean force because this conformational change is highly coupled to the binding of an ion in a different site here. And so we try to move the ion along the z-axis in the potential of mean force. And so when the channel is partially open, the gate is partially open, that's the activation gate, the filter tends to want to be around 8 angstrom, so that means it's conductive. This is the A conformation. And when you open the gate to, let's say, 23 angstrom, this conformation here becomes less stable, and now you go pinched. At the C conformation is more constricted like that. And this is the 2-dimensional potential of mean force, where R is the pinching coordinate for the two distances being average here, because we want to have just a single coordinate. And Z is the position of the highest ion relative to the filter. And so you can see that when you get pinched here in the A conformation, when you're conductive, the ions are in the site. This is around this position. This is like the site S2. like this ion here. And then when you get constricted, like for example in the C conformation, the ion is higher, and this is higher here. And if you collapse the potential mean force from 2 dimension to 1 dimension by integrating out the ion position, what you get is that as a function of the opening, there's really a 1-dimensional potential mean force. And when you're large, the conductive state is unstable and you go down to the pinch state. And then when you are 15 angstrom, the pinch state is accessible, but the conductive state is more stable. And so there's really a kind of a coupling between the activation gate and the filter. This is an allosteric coupling. Now, the process is happening too fast here because I told you the KcsA inactivates in about 1 to 3 seconds. And we observed that in half a microsecond. And so we were very disturbed by that for actually a couple of years. These results were obtained by Jared Osmeyer during his PhD thesis around 2014, 2015. And we were very, very puzzled. And it's only further work by Jing Lee, postdoc in the lab, that sort of shed light on this. Is that we believe that what happened is that when you give a stimulus, you go from the closed gate to a partially open gate. And when the gate is partially open, then the filter has a fairly high propensity to remain conductive. When you go to the fully open gate, like 23 angstrom, for example, then the transition to the inactive filter is so fast that by the time you reach this, you become inactivated. But it's this process here. You probably have many, many partially open states where this gate is flickering, in different state of partial openness that keeps the channel conductive for a while. And so we think that when you do single channel or population measurement, what you see is mostly this thing. That is the state that would be conductive. And this fully open conductive filter is a very low probability, short lifetime. And what convinces us of that is that if you truncate the KcsA has a C terminus, and this is C terminus here, as a C terminus is if you truncate that, you inactivate less faster. Here it's the black line. So when you have the C terminus, you activate to about 15% over a certain time. When you remove this C terminus, the channel tends to open larger, and it tends to inactivate even faster. And that means that because the gate is open more easily and unencumbered, you tend to inactivate faster. So this is our paradigm at this point. Why is also the inactivated state so long-lived? It's been a puzzle because, in fact, if you look at the constricted state and the conductive state, from the point of view of protein, the conformational change is very small. I mean, you talk about less than 1 angstrom RMSD on the backbone of the filter. So, a 1 angstrom RMSD of difference between 2 conformation normally would basically be obscured and swallowed by thermal fluctuation. So how can you get a conformational change that is so small and yet so long-lived? This is work that actually we have done before, but it was about the recovery. This is where we found that the role of these water molecules that bind behind the filter is actually quite critical, because if you try to make the channel conductive in the presence of those water, you get a clash, and the clash will make it so that it's impossible to recover. And if you do a simple kinetic model and you try to get the parameter of this kinetic Markov model based on molecular dynamics, you would say, oh, the rate of losing water molecules from one subunit, let's say, because let's say the 4 subunits are independent, is fairly fast. It may be 10 microseconds, you lose the water for 1 subunit. Of course, every time that you want to lose 1 subunit, it depends on how many subunits are there to lose their water. So at the beginning you have 4, then you get 3 or 2 or 1. And then the rebinding of water is actually very fast. It's about 80 nanoseconds. So if you have subunit that has lost its binding water, but they will rebind fast in less than 100 nanoseconds. So that means the probability to get this conformation versus this one, losing water versus not is only about 1.5% or something. It's not that large. And at the end, you bind an ion, in the filter, and that's when you fully recover. And so if you have 0 ion in solution, you cannot lock the filter in the conductive state. So you will hover over these 4 states, but you never lock the conductive state. So that's why it's dependent on ion concentration. And if you just simulate this simple Markov model with these fairly reasonable relaxation times, the binding of the ion is actually happening over 10 nanoseconds, so at 150 millimolar. And you can see that the recovery time will change from about 8 seconds to 16 seconds. And the colored dot are from the simulation of this Markov model, and the stars are from experimental data done in the lab of Eduardo Pirozzo. So, clearly what happened is that you can account for the long time just based on this kinetic model. And of course, if you change the osmotic strength of the external solution, for example, for sucrose, the recovery time will be a bit faster because now you have made it a bit easier to dry out that cavity, the waters that are bound in the cavity. And so even though you add sucrose and you make the system more viscous, you actually accelerate the conformational transition. So, having done that work, we felt pretty good about ourselves, and we were happy. But then, this became more controversial. Francis Velavitil actually pointed out that you can change the central glycine, where you get this pinching, by a D-alanine that somewhat prevents the pinching, and yet you still have inactivation in macroscopic current. But in fact, we inspected that by molecular dynamics, and it is true that if you have this D-alanine instead of the glycine, you cannot have the 4-fold pinching. What will happen is that you will get 2 subunits will pinch, but not the 2 others. And seen from the top, this is the potential of mean force of this pinching coordinate, and this would be the symmetric state here. Even in the wild-type channel, you have access to these kind of asymmetric pinching where 2 subunits would pinch, but not the 2 others. This is the conductive state. And so you see that you can get these asymmetric pinching. When you put the D-alanine there, you have no longer access to the symmetric pinch, but now you have access only to the asymmetric pinch, like that. And so that shows that, this particular chemical, this is an unnatural amino acid, so it's a chemical modification of the channel, can be accounted for microscopically. And then, of course, we still have only KcsA here. And most of these pores, like, for example, the shaker channel, the KV 1.2 channel, which was crystallized and is very similar to shaker. The MthK channel is another bacterial channel that has been crystallized to very high resolution. And the question is, well, how come we don't see any constricted conformation for any one of those channels? Since in principle, you can build, this is a homology model, you can see it here, 3 waters, and this is the conformation of a constricted shaker. This is a constricted conformation of KV 1.2 with the 3 waters here, and MthK constricted with 3 waters. And there's no, steric clash or anything. So in principle, it should be feasible, but the question is, this is a hypothesis, and this has been very puzzling that this is not observed. Very recently, Karina Nimijan and our collaborator, co-worker, I should name all of them, Céline Boiteux, David Poisson, and Toby Allen, did a very nice study where they were studying the MthK, and they looked at the binding of ions to the MthK experimentally with X-ray structure titration, and they also did computation. And their conclusion is that, of course, in order to pinch a filter, the ions need to get out of the way. And indirectly, the stickiness of the binding, how strong the ion bind to the filter, will affect your ability to capture and observe the constricted filter. It just turns out that KcsA does not hold on to its ion in the filter so strongly, not as strongly as MthK. MthK really binds its ions more strongly. They don't leave, and if they don't leave, then it's harder to access this constricted conformation. But computationally, they could show that if you actually have the constricted filter, as they show here, you will bind water behind the filter. So there really is, there are some answers to these kind of conundrums of why is it that MthK is not captured crystallographically in a constricted conformation. And so that's encouraging. Now, going back to Shaker, which is sort of the granddaddy or grandmother of all these channels where people studied inactivation, you want to know, can I account for inactivation for Shaker? Well, we don't have a crystal structure of Shaker, but there's a crystal structure of the Kv 1.2. And if you want to make the mutation of Kv 1.2 to turn it into Shaker, here they are. It's just a few residue, very, very few residue. It's not that many. It is almost identical. 87% of the pore domain is identical. So whatever you can learn from Kv 1.2, you should learn from Shaker. However, at the electrophysiological level, Kv 1.2 is known to inactivate considerably less than Shaker. So we decided to make the model of Shaker to capture this inactivated state in Shaker. And so we built a model of the inactivated state of Shaker where we transport the constricted filter from KcsA by homology and into the filter here. So this is a constricted, and this is our starting model. And basically, you can see here, you start constricted, and over a microsecond, the filter recovers. So it's doing the opposite of KcsA. If you start conductive, it stays conductive. And if you start constricted, it recovers to conductive. So failure here, no way to get a stable Shaker constricted. It's not because of a clash, though. And so having learned our lesson from KcsA, now we know that there's an allosteric coupling to the activation gate. And so this is basically the work of Jing Li to say, well, what if these are all KcsA structure with various degrees of opening? And you can see the gate can go from not very open to more widely open. So you have putatively some possibilities to look at more open gates. So what he did is take our model of Kv 1.2 and then just force it to go from the 23 opening to the 32-inch opening using targeted dynamics. You just slowly generate models where the gate is progressively opening. And then he collects models along the way to try to see if these models will be stable. And there we got finally a model where this Shaker structure will have the water behind the filter, not 3 water, but only 1. There are waters that come around here, but they're more labile. And they come in and they come out much faster than in KcsA. The only water that seems to be really bound with a long live is the bottom cavity here. And then the filter is really now, you can see, stable in this pinch structure like that. So it is truly constricted and very stable. This is, I highlight the time scales. This is stable over 20 microseconds now. But this is a long simulation, and it's been stable like that with no doubt. This is 20 microsecond simulation. And once you have that, again, it's nice to be able to go back to a more quantitative assessment. And you can see that when you have a channel that's open, like this is basically like the Kv 1.2 crystal structure, right? So this is 18 angstrom. The conductive state is more stable. This is more stable. You would have only a metastable state here. You'd basically go back to the conductive, which we observe in our molecular dynamics. Now, if you open to 32 angstrom, like we did in our model, now the constricted state is the most stable. You could start conductive, and it basically will make a transition, and presumably this would be more stable. With the same amount of opening of the activation gate, KcsA, has more propensity to inactivate than shaker. You can see this is no, we cannot go there, but KcsA can. And this is actually true. KcsA inactivates fairly deeply. Shaker inactivates, but quite a bit less deeply than KcsA. I think KcsA is more like 10% the depth of inactivation at the of wild type KcsA. The depth of inactivation of shaker may be more like 50%-ish, 60, 50%, maybe 40%. It's hard to, it depends how it's being measured, but it's not as deep. One thing we observe actually when we compare the constricted structure of KcsA with the shaker, it's very fascinating, is this is the water molecule at the bottom of the cavity, that's and this is the constricted filter. And you can see here, the carbonyls are going to the left. So if you go donor acceptor, it would be in this direction. This is donor acceptor. Shaker is actually the opposite direction. It's fascinating. And the most immediate residue in the neighborhood of this water is the glutamic 71 in KcsA, which is a valine in shaker. And it's a valine actually in KV 1.2 also. In most voltage gated channels, it is a valine, but in KcsA, it's a glutamic acid. If you put a valine there, you can revert, this thing and make it shaker-like in KcsA. So this is a fascinating thing, and I want to go quickly here, but you can look at the handiness of this thing, whether you go one way or the other, and this is the dihedral of the handiness, like that. And in KcsA wild type, this well is a little deeper than this, but the other handiness is accessible, Of course, in shaker wild type at 18 angstrom, you're only conductive, and these two states are super high energy, but the other handiness would still be more accessible. If you go to the 30 angstrom or 32 angstrom, conductive is less stable. Now you have this state that is stable. This is inactivated, but this is this handiness. In KcsA E71, this is a little bit more stable, but you would still be able to go to the other guy. So this handiness of the pinching is a little quirkiness that deserves some attention. There is a very famous, oops, I have to show this here. There's an absolutely famous mutation in shaker that's been studied since it's the 434 F mutant. It's a phenylalanine. If you change a tryptophan into a phenylalanine in shaker, and it's just around here close to the filter. If you put a phenylalanine there, the filter is known to inactivate very, very fast. In experiments, it's been very difficult to characterize that structurally, but we can do this in molecular dynamics. And what we see is that indeed the filter would become non-conductive, but it's actually more non-conductive in an outer side. It's not the same constricted conformation. This is more like close to the S1 side. It's not the S2 side. And it's also, it bears some asymmetry a little bit. You see, the distance is not quite symmetric between the two monomers. So even though this is often presented as being a mutation that makes channels constitutively inactive, the nature of the constricted conformation may be different for this mutant. Now, let me sort of quickly wrap up on the Herg channel. This is something that so the Herg channel, there's no crystal structure. There's a high resolution insofar as electron microscopy structures go by Rod McKinnon. So it's about 3.8 angstrom of the Herg channel. The Herg channel is very important because it's in the heart, and it has a lot of importance for cardiac action potential and the function of the heart muscle. If you, the Food and Drug Administration has to test any drug for anything, many drugs for inflammation, whatever, if the drug will bind and interfere with the Herg channel. Many drugs bind to the Herg channel and actually enhance the inactivation of the Herg channel, and that will cause arrhythmia. And if you bind to the Herg channel, it's basically kiss of death for your drug program. It's gone down the tube because this is such an adverse effect that you cannot put this on the market. You would kill the patients. There's several mutations of Herg that are known to affect inactivation. And what can we do about Herg from the point of view of molecular dynamics? So this is a paper that just appeared basically a week ago online last Friday. And what we did is, again, take the Herg channel from Rod McKinnon and run long molecular dynamics simulation. This is, we had like a 15 microsecond simulation. What you see is that the channel will pinch, but it will be an asymmetric pinch spontaneously. We did that a few times. The asymmetric pinch is reproducible and happens sometimes faster than other times, but it's asymmetric. So that means two subunits will be kind of constricted like, two subunits are not so constricted. So you get an asymmetry like that. In the EM structure, this is the EM structure here, the density from EM, and in the red is the EM structure, is fourfold symmetric. And so our result departs from the EM structure, which is done, in single particle and all that. So you could try to argue that maybe the EM structure could be refined with a two-fold symmetry rather than a four-fold symmetry. But in fact, that's not so true. This would be the density that you should see. And this density that predicted from the modeling is a bit different. So this is a kind of unresolved question. Exactly why is it that the cryo-EM did not capture that asymmetric conformation? But nonetheless, we did do that. And this is, again, the PMF versus the two distances. And you've seen PMFs, I guess. So these are the asymmetric constriction. There is no symmetric, there's no symmetric constriction of Herg. A bit like with the D-alanine of KcsA. So this is something that we have seen before. Again, if you compare KcsA, Herg, and Shaker. Oh no, this is just to remind you. So KcsA wild type can have the symmetric constriction, but it's not here in the D-alanine G77, and it's not there also in the Herg. So this asymmetry is something we've seen before. Now we decided to study several of the mutation in Herg to try to beef up our contention that we're capturing something that's meaningful. And so this is the wild type inactivates to some degree. This is a mutation that's known to inactivate more. And then we did electrophysiology to actually also confirm that. I mean, like there was published electrophysiology before on most of these mutants, but we just wanted to have uniform conditions on everything. So you can see here, conductive to constricted is enhanced by that mutation. And that's because there is a hydrogen bond between this tyrosine here and serine 620. So when you change the phenylalanine into a tyrosine, you have a new hydrogen bond here that stabilizes the constricted conformation. If you put the threonine there, you actually stabilize the conductive state. And that's because now you have, instead of a serine here, you have a methyl group that's in the way. It pushes the phenylalanine out of the way, and you're more conductive. Interestingly, if you have the tyrosine here, but you have a threonine, you cannot form the hydrogen bond anymore because now the methyl group is in the way. This result was not published before. This was a prediction of the computation that was then verified experimentally. And so this is an absolute prediction of the computation. There's another one also. There's an asparagine 629 that forms this hydrogen bond here on another tyrosine. And if you put the aspartic acid there, you really stabilize the conductive, in part because you probably have created this hydrogen bond here. You've created Well, first of all, also you've recreated the position that is the D in most of these other channels, and you will have a water molecule that will be here, and that will also stabilize a conductive state. Herg inactivates more than KcsA, which inactivates more than Shaker. So if you have the same degree of opening on all of those channels, then Herg would be fairly conductive. KcsA would be conductive with some access to the inactivated state, but Herg would be mostly inactivated. And so this is actually true to experiment, that inactivation is more pronounced in Herg than in KcsA and more pronounced than in Shaker. There is a mutation where people insert a disulfide close to the outer mouth of Herg, and that completely prevents the inactivation. And we ran the simulation, so over like 5 microseconds, you could see that you stabilize a conformation very, very quickly, because the disulfide is a pretty stiff thing. You stabilize a conformation, but it's not really the canonical conductive conformation, because you have widened the outer side, or the extracellular side of the pore. And so this filter would be conductive. The prediction would be also that this filter would be losing some selectivity, because selectivity is a process that requires a single file of ions, and now you don't have a single file of ions. So this is in part why this channel will conduct, this mutant will conduct. It's locked into a conductive state, but it's not the same conductive state. So it's a bit like the W434F, where you have to be a bit cautious. Some of these mutations don't lock the canonical conformation that you're after. So you just have to be a little bit cautious about that. So to summarize, we believe that inactivation is a process that is related to a constriction-like conformational change of the filter. It may not be, I call it constriction-like because they're not completely identical. There could be a difference even when you make a D-alanine in KcsA versus the wild type KcsA, or whether you put the E71 in KcsA, E71V versus wild type. So there are some subtle differences, but by and large, we're talking about a narrowing of the filter to the point where ions cannot conduct. And of course, it's affected by the stability of the filter, the conductive filter. And if you make mutation close to the top of the pore domain, you will affect inactivation. The nature of the permeant ion, like if you change the permeant ion from potassium to rubidium to sodium, but sodium is not totally permeant, or cesium, you will affect the extent of inactivation, because the filter has to go from conductive to non-conductive, and that is definitely affected by the balance of binding of ions to the filter. So that is not shocking. It is going to be a difficult effect to tease out, because you have to really, at least from a computation point of view, describe the interaction of ions with the filter with very high accuracy. So that's going to be a difficult thing to tease out. There is clearly an allosteric gate filter coupling. That means that the amount of opening of the activation gate talks to the filter. I didn't say exactly how this happens, but there's a few bulky residue in this area that if you make them smaller, like an alanine or something like that, you sort of diminish very, very much this allosteric coupling. And then in the case of KcsA, that means you almost relieved the inactivation. And so that's definitely something that you can watch out for in different channels. And in Shaker, for example, this is not a very large residue. If you substitute there the large residue that we have in KcsA, you actually increase the allosteric coupling. And so this is something that has definitely an effect. There's also some direct effect of the voltage sensor to the outer side of the pore domain that can have some effects. And so there's mutation sometime in the voltage sensor that will directly affect inactivation. Some of it could be also through the linker and the allosteric gate. Some of it could be through space. And if you change also some things about the lipids or what is membrane constituent, you could through space affect what's happening to the pore domain. So there's different families of effects, and they all sort of talk to each other. So, doing this experiment is extremely hard to dissect precisely. And so you have to be very cautious with, drawing unambiguous conclusion. I don't want to read all my conclusion here because it's essentially a summary of the talk, but I would say the constricted filter is a plausible, constricted-like or constricted filter is a plausible model for C-type inactivation. Asymmetric constricted seems to be a recurrent theme. And that's about it. I'd like to just maybe end by thanking many of my collaborators. A lot of this work was started by a very, very bright graduate student, Jared Osmeyer, who's now at Texas A&M as assistant professor. My absolutely beloved collaborator, Eduardo Peroso, also Pencho Bezania, has very, very much influenced all my work and my thinking. Experimental collaborator that are extremely valuable: Chris Ahern, Marcus Weingart, Sujosha Kapani, Luis Cuello, and then people in my lab who work on that. Jing Lee did a lot of the work. Young-Hoon is a graduate student working on this right now, and Ahmed Rim is a postdoc working on that. Some work was done by Albert Pan also, who's now at the Shaw Research. So I should
Benoit, thank you very much for this inspiring talk, which
stop share the screen now. I can answer questions if there are some questions.
Yeah. So
perhaps not questioned in Italian though. It looks like some comments are written in Italian.
Are they?
I see. Yeah. I mean, I could read them, but I don't think I can understand.
So the first question would be from an Italian, but in English, indeed. And
sure.
That would be from Luigi Catacuzzeno. Does the rate of inactivation have any dependence on the filter content in terms of potassium ions and water molecules? What if you use a water-devoided filter?
Okay. Well, so most of the time, in a few cases, I could show spontaneous conformational change in my simulation. In general, the spontaneous conformational change was one way. Like the KcsA would go from conductive to constricted over, let's say, a microsecond. In the case of the shaker, we would see also spontaneous simulation. So generally, that would mean that by and large, the conditions of the system were such that the initial conformation was really metastable and did not have a long lifetime. In experiment, you would probably be more at the edge of, so that means that when I see only a one-way transition, I don't really know the rate. What I have is only a one-way transition. It's not a rate. And the true rate experimentally would probably be something that is quite complicated to tease out. For sure, it must depend on every obstacle and every interaction that is present from the reactant to the top of so-called free energy barrier transition state to the the product on the other side. And so from a computation point of view, it will depend on the content of the pore. It will depend on the strength of the interaction of the ions and the water. If the water was not there, these interactions would be absent. If water was interacting more strongly behind the filter, then you would recover more slowly. That's for sure. These rates will be affected. Rates are going to be affected, and even the equilibrium population will be affected, probably.
So there's a question now from Sergei Shukarev, who asks, is the rate of inactivation in these channels slower in the case of very high potassium concentrations?
Yeah, in KcsA it is slowed down, and the depth of inactivation is relieved by potassium. Yeah.
And then one from Lucie Delemotte, who asks, what is your best guess about why the Herg structure was captured in the non-pinched state?
You know, it's, I don't want to say a lie here. What was this done with nanodisk with lipids, or was this done with detergent? I think it was a structure done with detergent. If somebody can send you a comment, I don't want to say something erroneous. I believe it was done with detergent. And so it clearly is not a channel. So there's definitely two things. It clearly is not a channel in a bilayer. If it's in a nanodisk, that would be like a kind of a model bilayer, but it's even that would not be a real bilayer. The other thing is that it is a fast frozen structure. And so if you have a balance of entropy and enthalpy in a molecular structure, when they fast freeze these structures, the hope is always that they fast freeze them so fast that they quench, you know, the snapshot that is present at the room temperature. But the cooling rate is never that fast, that if you have a process that can occur in some microsecond or so, probably even millisecond, that the protein could interconvert. So it's possible that the balance of enthalpy and entropy such that when you fast freeze, you know, you go back to a fourfold symmetric. So there's different physical factor about this. With Eduardo, we reanalyzed a little bit the EM data to see what if you did not enforce fourfold symmetry. Would this twofold symmetry pop up? And it does not. So from the point of view of just the pure analysis of the EM map, this is not an oversight by imposing fourfold symmetry. So there's something deeper going on that is probably as with the physical status of the sample itself, probably.
Yeah, so indeed, Lucie was following up on her own previous questions, saying that symmetry plays a role, but I believe that you have already partially answered to that. I don't know if you want to add something.
Symmetry
plays a role.
Yeah. I'm not so sure how to interpret what she says here, but the you know, because you have 4 monomers, it's not clear.
So
every dominant, it's just because you have 4 monomers that are chemically identical, it's not clear that all metastable state of the system have to be fourfold symmetric, obviously. And it's not even clear that the ranking, the free energy ranking of the population of those metastable state has to be affected by symmetry in an obvious way, you know. Yeah. I know that in quantum mechanics, if you have a double well potential with a barrier and you talk about the wave function, you know, of a two-level system like that, the symmetry of the Schrodinger equation is such that you can deduce the ranking of your eigenvalues based on the symmetry of the problem. But I don't think that with a molecular structure like that, from the point of view of statistical mechanics, this is actually a preordained condition. So it could be that the more stable inactivated state is just, you know, asymmetric like that. It could be.
Okay, so Lucie says that you have answered, and she thanks you. There's a comment by Karina who says that the Herg structure is in detergent, and I would like to add a question for you, whether to finish with this beautiful final lecture. You have a way to connect the Herg susceptibility to so many drugs with these inactivation mechanisms. So in some way, if the pockets behind the selectivity filter play a role, if you have binding some suspicious sites, which you have shown that are involved in this mechanism.
Again, maybe Krina or somebody can comment. You know, of course, there's the EM structure of Rod McKinnon of Herg, And I am certainly aware of a number of people who are continuing to do Herg structures with EM. I'm not so sure, perhaps there have been publication of other EM structures with drug bound. Of course, drugs are not very large molecules, so at the main thing you would get from an EM structure, unless you have like a super high resolution, would be at least the general location of where the drug binds. In some cases, you may even have a hard time locating where the drug is bound. We would like to study the binding of drugs to this Herg inactivated state. It's a bit risky unless you know the general area where they bind. It's definitely risky because it starts to be speculative. We'd have to do the docking and then maybe do some free energy calculation or something like that. You know, this would be, it's certainly of interest, but it's an undertaking that has some risk, because if you dock in the wrong place, or even if you dock at the right place but in the wrong orientation, you know, the work that you do is not as meaningful. And you know, this is, I think, you know, I wanted to say that, but maybe say that even for the next, the discussion, the panel discussion. I think, you know, we're at a stage where, this was done with the charm force field. This is a non-polarizable force field. Charm force field, a lot of the simulation were done with NAMD, some with Anton, some, I think some with OpenMM even. Or Amber GPU. So we're beginning to be pretty agnostic about the software in my lab, I would say. And, you know, I would say that by and large, what comes out of those simulations, given the initial condition, is not unreasonable. But you start from initial condition and you discover the metastable state that are kinetically accessible within the range of the time scale that you simulate. And then if you do umbrella sampling and things like that, you're going to get, you know, relative population between these different accessible metastable states. But if something is really beyond the time scale that's accessible, then it's not really a failure of the computation. It's just, it's more like the lack of ability of getting there, you know. So I think the caveat is more like, it's not that we get garbage, is that you get within the neighborhood of what you put in, and you explore in that neighborhood. But if you go too far and you have no chance to discover whether, you know, what you said is true, like, for example, if you dock a drug in the wrong pocket and you simulate only 10 microseconds, maybe it needs 100 microseconds to leave that pocket, then you're not going to know that this is wrong, and it's going to be a bit of a waste of time. So, you know, that's so I think, you know, that's why I'm always a bit prudent about venturing a little too far off the reservation. I try to remain at least within the, you know, within sight of what was established experimentally, and try to just expand the perimeter a little bit, because otherwise it's so speculative that you could just waste your time. The danger is that you could waste your time.
Benoit, thank you very much. I think also this leads to a nice question for the following roundtable, which is how to make an educated guess to start our simulations, which is more or less what you have better said. So with that, I would like to thank you very much.